Now let's move on to discuss the cephalosporin drugs. These are beta-lactam drugs that also inhibit cell wall synthesis by binding transpeptidases and interfere with the peptidoglycan cross-linking. Like penicillins, they inhibit bactericidal activity, but luckily are less susceptible to beta-lactamases. The penicillins are grouped into 5 generations. First generation is composed of cefazolin and cephalexin. Second generation, cefoxitin, cefaclor, and cefuroxime. Third generation is ceftriaxone, cefotaxime, ceftazidime. The fourth generation includes cefepime and ceftaroline. I know these are all similar sounding drugs, and this can be frustrating memorization for some students. It is important to remember that on the boards, they're not going to ask you which of the following is a third generation cephalosporin, but it is important to recognize their names and what generation they are in order to appreciate which bacteria they have some activity against. Remember, you just need to recognize them in the stem of a question. As the generations increase from 1 to 5, the drugs have better coverage against gram-negative bacteria. The first generation cephalosporins mainly cover gram-positive organisms, while the 5th and 4th generation can also impact gram-negative organisms. Before we go through each generation individually, the toxicities include a cross-reactivity with penicillin, hypersensitivity reactions, hemolytic anemias, a disulfiram-like effect, vitamin K deficiency, nephrotoxicity with the aminoglycosides. Thinking back to basic biochemistry, why do you think we would see a vitamin K deficiency? Remember, the absorption of vitamin K occurs in the gut. In order for it to be absorbed, it must be modified by enteric bacteria. Remember, enteric stands for gut. So in order for vitamin K to be absorbed properly, it must be modified by these gut bacteria. When we give these drugs, the enterobacteria are actually killed, and this reduces the vitamin K modification and then results in a poor absorption of vitamin K and resulting in the deficiency. Cefazolin and cephalexin are the first generation cephalosporins. These guys are effective against gram-positive cocci. They are narrow spectrum and bactericidal, and we can see the sniper rifle again. However, they do have some activity against gram-negative organisms, including Proteus, E. coli, and Klebsiella. We can remember these bacteria by remembering the mnemonic PEC, Proteus, E. coli, Klebsiella. In general, these drugs, the first generation cephalosporins, are only effective against gram-positive cocci and 3 gram-negative rods. Cefoxitin, cefaclor, and cefuroxime are second generation cephalosporins. These drugs are similar to the first generation cephalosporins in that they really just cover gram-positive organisms. However, they have increased activity against gram-negative organisms, including Haemophilus influenza, Enterobacter, Neisseria, and then the Proteus, E. coli, and Klebsiella like the first generation, and then also Serratia. So this second generation of cephalosporin has activity against 4 more gram-negative bacteria than the first generation cephalosporin. We will see this trend continuing as we continue to go through the later generations. Ceftriaxone, cefotaxime, and ceftazidime are third generation cephalosporins. These are very powerful drugs that are used to treat gram-negative infections. Ceftriaxone is a first-line agent used in treating Neisseria meningitidis, Neisseria gonorrhea, and Lyme disease. Thinking back to bacteriology, do you remember which bug causes Lyme disease? That would be doxycycline. Very good. Also of note, ceftazidime is active in the only third generation cephalosporin that has activity against Pseudomonas. Cefepime and ceftaroline are the 4th and 5th generation cephalosporins. Cefepime has activity against Pseudomonas, and ceftaroline has activity against MRSA. Ceftaroline, as a 5th generation cephalosporin, essentially ceftriaxone plus MRSA coverage. It covers everything ceftriaxone does, including the Neisserias and Borrelia, and also MRSA. This is the only cephalosporin that covers MRSA. Another important thing to know for step 1 is the mechanism of resistance bacteria have against cephalosporins. Thinking back to the penicillins, what was the major form of resistance? Remember, for the penicillins, it was the beta-lactamase enzymes. Fortunately, for cephalosporins, beta-lactamases aren't very active against these drugs. The mechanism of action of resistance for the cephalosporins is different, and it is an altering of the penicillin binding protein. The penicillin binding protein is the transpeptidase, So the bacteria, what they do is they change the structure of the transpeptidase enzyme so the cephalosporins cannot bind. What two cephalosporins act against Pseudomonas? Ceftazidime and cefepime. Very good. The carbapenems are the drugs that end in enum. They include imipenem, meropenem, ertapenem, and doripenem. These are beta-lactam drugs that are resistant to beta-lactamase enzymes. These are powerful drugs with a very broad spectrum. Remember our army guys? They have activity against gram-positive organisms, gram-negative organisms, and anaerobic organisms. They are considered the big guns in the hospital and are typically saved as a last resort to fight very resistant organisms. These drugs are administered with cilastatin. Do you remember why? These drugs are inactivated by an enzyme called dihydropeptidase 1 in the renal tubules. Cilastatin protects the enzymatic inactivation by this enzyme. The mnemonic to remember this is the kill is lastin with cilastatin. One of the reasons these drugs are reserved for particularly resistant organisms is because they can cause a very bad side effect profile. They can cause GI distress and skin rash, but more importantly and scarily, they can cause seizures at very high levels. Now let's talk about the aztreonam drug class. The aztreonams are a unique class of beta-lactam drugs, known as the Monobactams. The word monobactam describes the chemical structure of the drug, and it basically just means that it is the beta-lactam ring by itself that is not connected to this 5-membered ring here. The beta-lactam ring is the same and therefore has the same mechanism of action as the penicillins, carbapenems, and cephalosporins, but this change in structure also makes them have less susceptibility to the beta-lactamases. Another benefit of the unique structure of aztreonam is that it is not cross-allergenic with the penicillins. So what do I mean by that? Well, basically, it means that even if a patient is allergic to penicillin, aztreonam is safe to use because there is no cross-allergenicity. This is an important thing, and it is why this drug is used in patients with renal insufficiency and a penicillin allergy. It tends to have few side effects. However, unfortunately, although it has many benefits, aztreonam is limited in its utility and is only effective against gram-negative rods. What 3 gram-negative rods do the first-generation cephalosporins have activity against? Remember, the cephalosporins mostly have gram-positive activity. However, the first generation is active against 3 rods. Do you remember what they are? They are Proteus mirabilis, E. coli, and Klebsiella. You can remember that with the mnemonic PEC. Now let's discuss the drugs that interfere with peptidoglycan synthesis, most importantly, vancomycin. Vancomycin is not a beta-lactam drug, it doesn't have a beta-lactam ring, and therefore it is resistant to the beta-lactamase enzymes. Vancomycin binds to the dealanine residues of an essential protein in the bacterial cell wall, therefore blocking its synthesis. You can remember this by thinking of 2 dollar bills shown here. It is key to know that vancomycin is only effective against gram-positive organisms. It is particularly effective against MRSA, Staph epidermidis, Enterococcus, and C. difficile. The most high-yield portion of this drug is to know that it is effective against MRSA. Vancomycin is well tolerated and it is widely used. However, it is not trouble-free. The main toxicities associated with vancomycin include nephrotoxicity, ototoxicity, and thrombophlebitis. Do you know what thrombophlebitis is? Well, to understand what thrombophlebitis is, let's look and break down the word. Thrombo also means thrombus or clot, and phlebitis means inflammation of the vein. Itis means inflammation, and phleb is short for vein, or is another word used to describe the vein. So, vancomycin causes thrombophlebitis, or causes clot formation in a vein that causes inflammation. More common toxicity associated with vancomycin is what's called red man syndrome. Red man syndrome is common when the vancomycin is given in a rapid infusion. The syndrome is characterized by red flushing after administration of the drug. In response, what would you do to prevent this or stop this from happening? Well, first, you would stop the infusion of the drug, and then you can give an antihistamine, such as Benadryl, and cause the reaction to stop happening. Unfortunately, bacteria are able to develop resistance against vancomycin. If we remember back to the mechanism of action, vancomycin binds to the dealanine residues on an important protein found in the cell wall. The way the bacteria become resistant to vancomycin is by converting one of the dealanine residues to a D-lactate residue. By doing this, vancomycin is unable to bind to the protein in the cell wall and therefore is unable to stop the synthesis. This is a very important mechanism of resistance and one that is highly tested on step 1. Now let's spend some time discussing the medications that target protein synthesis in bacteria. But before we talk about each drug individually, let us review some aspects of protein synthesis. Thinking back to basic cell biology, do you remember what organelle is responsible for forming polypeptide bonds? That would be the ribosome. Very good. And do you remember what the different sizes of human and bacterial ribosomes are? Remember, human ribosomes are made up of 60 and 40S ribosomal subunits, while bacteria have 50 and 30S bacterial subunits. An easy way to remember is that the first digit in the bacterial ribosomes are odd, and bacteria are odd organisms. Our drugs that target protein synthesis specifically target the bacterial ribosomes, 50S and 30S, and leave the human subunits alone. Here is a wonderful image from first aid that details the process of protein translation. Translation starts with an mRNA molecule binding to an initiator tRNA molecule. This forms the initiation complex. Once the initiation complex is formed, peptidyl transferase comes in and forms the peptide bond between the amino acid residues from the tRNA to the elongating polypeptide strand. So next, the elongating polypeptide strand is translocated and prepared for the next amino acid addition. Each step along the way is targeted by one or more antibiotics. Overall, there are 6 classes of protein synthesis inhibitors. We have the aminoglycosides and tetracyclines that inhibit the 30S ribosomal subunit. And then we have clindamycin, chloramphenicol, erythromycin, or the macrolides, and linalazid that inhibit the 50S ribosomal subunit. A great mnemonic to remember this is that buy at 30, sell at 50. To begin our discussion of the drugs that inhibit protein synthesis, we will start with the aminoglycosides. The aminoglycosides include gentamicin, neomycin, amikacin, tobramycin, and streptomycin. A nice way to remember these drugs is to remember the first letter of them form the word NATS. The names can get confusing because in addition to the aminoglycosides, the macrolides and vancomycin end in the suffix mycin. So I would try to remember these names so you will be able to distinguish them from vancomycin, erythromycin, azithromycin, and clarithromycin. Aminoglycosides inhibit the formation of the ribosomal initiation complex and cause misreading of the mRNA. We can remember this by knowing that A in the beginning of aminoglycosides initiates the alphabet, and that will help us remember that aminoglycosides affect the formation of the initiation complex. There are 2 important factoids about aminoglycosides that the boards like to test you on. One is that they require oxygen to uptake into cells. That makes them ineffective against anaerobes. Also, they are synergistic with beta-lactam antibiotics. Synergism means that when used together, aminoglycosides and penicillin are more effective than just when they are used alone. Instead of 1 + 1 = 2, 1 + 1 = 3. In practice, aminoglycosides are given with penicillins. For instance, gentamicin is given with ampicillin for neonates with fever and a suspected infection. These drugs are bactericidal and have a narrow spectrum. See our sniper guy again. They are only effective against aerobic gram-negative rods such as Pseudomonas and E. coli. Just like vancomycin, aminoglycosides are not trouble-free. In fact, they are notable for their nephrotoxicity, and that is worsened when they are co-administered with cephalosporins. They can also cause ototoxicity, as well as they have teratogenic potential. The final N is neuromuscular blockade. In terms of resistance, bacterial transferase enzymes inactivate the drug by acetylation, phosphorylation, or adenylation. Now for a flash quiz. What beta-lactam drug that we've talked about can cause seizures? Remember, this is an important side effect, which makes us not want to use the drug. Very good. Imipenem, or the Carbapenems, are notable for their ability to cause seizures in patients. Remember, these drugs are big guns, and they are reserved only to be used in organisms that are highly resistant to a number of different antibiotics.